Hi everyone, welcome all. Thank you all for coming to the Medical Sciences monthly lecture. Right now we have about nearly 200 attendees, which is really exciting. These sessions are designed to inspire you into the world of medical sciences, and we appreciate you being here with us to explore gene and stem cell therapies with Professor Emma Morris, which is a hugely interesting topic for anyone interested in medical sciences. My name is Anisa Chowdhury, and I study nutrition and medical sciences here at UCL, which delves into the science of nutrition and the impact it has on the human body. I will hand over in a minute to Emma, but before I do, please do pop any questions you have in the chat box. After the lecture, we will have an opportunity to ask Emma these questions. No questions are too big or too small, and we will try to get through as many as possible. I will now introduce Emma. Emma is a clinical, academic, and international leader developing novel gene and cell therapy approaches for the treatment of cancer, infection, and immune disorders. Her major focus is on leading the expansion and adoption of advanced therapies and stem cell therapy transplantation into routine clinical care for NHS patients. Emma was elected a Fellow of the Academy of Medical Sciences in 2020. She will now begin her lecture.
So it's a real honour to be able to talk to you all today. Thanks for the brilliant introduction and welcome to all of you. I've got 3 teenagers at home, one in year 13, one in year 11 and one in year 9. So I hope for some of you, if you're doing biology GCSE or biology AS or A levels, this will be a bit of helpful revision, but also good fun. So as Anisa said, I work both as a doctor looking after patients doing stem cell transplants and gene therapy, but I also run a research laboratory at UCL developing new forms of gene therapy. So we're going to start with a bit of biology revision. You all know that humans are very complex machines. Each one is made up of 37 trillion cells. So that's 37,000 billion cells. Each cell in our body does a different job, with every organ of the body having its own very specialized cells. For example, if you take a cardiac myocyte, a heart cell, and grow it in the lab in a dish, it will start contracting, even though it's not part of a formed heart muscle. And if you take immune cells and grow them in the lab, you can see that they will leak toxic chemicals whenever they encounter bugs or danger signals. That's how they manage to kill pathogens. Don't forget that every individual cell is made up of billions of protein molecules. So if you think this is a picture, you'll be familiar with this picture of a eukaryotic cell. If you think of the cell as a house, the proteins are all the things in it that are like really useful. They do stuff or they make things work. For example, if you think of a house, proteins can form the staircase, the furniture, or the equivalent of enzymes in a cell in a house would be the electricity or the water supply. So if you think of that, basically cells can only do their job because they're made up of protein molecules. Now within the nucleus, I don't know if you can see my cursor, that's where all the genetic material is present. And it's organised in a very special way. And you know this, it's packaged together in chromosomes. But if you look more carefully, the DNA is formed into a double-stranded helix, which is all ravelled up. And it's wrapped round and round proteins called histones. And these are then ravelled up together very tightly into chromosomes, which all sit within the nucleus. And it basically, the DNA is like the script for a play. And together with all the producers and the directors, it provides instructions for the actors to actually produce something. Now, an important thing to think about is the fact that this is a double-stranded helix, and there's a real purpose to that, because it allows the DNA double strand to break apart so that it can be copied. And this is really important because every day we need to generate new cells. It's critical to life. Without the ability to do that, we would die. Now, every single second, 1 million cells in your body die. Every single second. And that is 1.2 kilograms worth of cells dying every day. Now, I think that's quite a mental fact, right? And the only way we survive and the only way our organs carry on working properly is if every time a cell divides, it's replaced by an identical copy. Now, you know from your school biology lessons that DNA is transcribed into mRNA, and the mRNA is translated into proteins. And this is required for whenever a cell divides, it needs to make more protein, so it increases in size and then it splits into two. So not only do you need to copy the DNA, but you also need to make more protein. Otherwise, every time a cell divided, it would become half the size and they would get tinier and tinier and tinier, and then they'd end up being nothing. So we're nearly at the end of our basic biology revision. But one final thing, which is an interesting fact about RNA, is that the Pfizer and the Moderna COVID-19 vaccines are made of RNA. So not many of you will have had your COVID vaccine yet, but some of your older relatives may have done. And this is a cartoon drawn by a friend of mine called Pedro, and he does lots of science cartoons. He also works as a scientist. But it highlights how the Pfizer vaccine works by introducing foreign RNA, which is this pink strand here in the cartoon, into cells. And the cell's own ribosomes, which are shown as these brown blobs here, actually sit inside the cell, and every time messenger RNA comes along, they translate the messenger RNA into protein, and that's their job. They've been doing it for years. So you can see here the ribosome saying proline, methionine, asparagine, and stop when it reaches a stop sequence. And then it says, right, what's the next bit of messenger RNA? I've got to make another protein. This is the entire job of the ribosome. And these two ribosomes obviously are talking to each other. Oh, I don't think that one came from the nucleus. And this pink ribosome is actually from inside the cytoplasm, and it's got there via the vaccine. But anyway, the ribosome doesn't care. The messenger RNA goes through the ribosome and is converted into one of the proteins of the SARS-CoV-2 virus. So our cells are making viral protein. And of course, it's that viral protein that is then recognized by our immune cells, which then expand and are able to kill the virus and protect them from it. Now, proteins, of course, carry out, as I said before, most of the functions of a normal cell. And that's true for our own proteins, but it's also true of viral proteins. So, in fact, the spike protein that's made inside our cells when we have an RNA vaccine against COVID is actually binds onto a human cell receptor, and it allows the virus to get inside the cells. So it's an important functional part of the virus. So let's summarize what we've learned about DNA, RNA, and protein. So the DNA, or the genetic sequence itself, is really a bit like a book. It's a whole description, a big long story. It's all the different things that can be made. Now, when we think about the human genome, there are many, many genes, and not all genes are switched on in all cells. So it's a bit like you dipping into a book of short stories, for example. You don't have to read all the chapters, or you don't have to read all the stories, but you need to read the instructions that allow you to do what you're going to do. So if this was a recipe book and you want to make a cupcake, which we can equate to the protein, the book, the whole genetic sequence is the recipe book, and the RNA is the actual recipe for the cupcake. And then if we think about the cells and our tissues, that's basically a collection of products together. In this case, it's a cake shop. So that's a little bit about DNA, RNA, protein and cells. I also work on stem cells and carry out stem cell transplants. And stem cells are a very special type of cell. You're born with them and they survive in your body for the whole of your life. One of the really important properties of a stem cell is that it's capable of dividing and self-renewing indefinitely. So they basically give rise to daughter cells. They can self-renew. They're very unspecialized, but the daughter cell can go on and become many different types of mature cells. And that's a process in biology that we call differentiation. Now, there are a number of different types of stem cells. Broadly, there are 3 types. There are embryonic stem cells. So these are the very, very early cells in an embryo that basically, if you start off with a 4-cell embryo, they give rise to all the different cells of your body and all the different organs in your body. So they are truly pluripotential, which means they can become anything. There are also cells called adult stem cells, which are not pluripotential. They can't become hair and nerve and skin and cardiac tissue and immune cells, but they can become a number of different types of immune cells or blood cells, for example. And we call these multipotent stem cells. And an example of these are hematopoietic or bone marrow stem cells, which I'll talk about in a bit. So the embryonic stem cells can become all the different cell types, and the adult stem cells can become a number of different cell types with very different function. And then finally, there's another type of stem cell called induced pluripotent stem cells, or iPSCs. These are really neat. They're artificial cells that have been reverse engineered in the lab. So an example is if someone took a skin biopsy of me and had cleaned the skin so it was sterile, grew my skin cells in a dish in a lab, and then put various growth factors on the skin cells so that the cells would then revert and go back to a less differentiated state, so become a stem cell. So go backwards, basically. It would be a bit like taking one of you and turning you into a toddler or a baby over the course of a few days, and then culturing the induced stem cell to become a different type of cell. So it's a way of turning a skin cell, for example, into a hepatocyte, which is a liver cell. Now, this at the moment is being done in research laboratories, but in the future, it has the potential to allow us to generate new tissues and new organs from individual patients to use for transplantation or regenerative medicine, rather than using a donated organ from someone who has unfortunately died. So more about hematopoietic stem cells or bone marrow stem cells. So I'm one of the bone marrow transplant or stem cell transplant consultants at UCL. And you can see in the middle, if you can see my cursor, there's a cell called an HSC, or a hematopoietic stem cell. You don't need to understand the details of this, but you need to see that it can become all sorts of different cells of the immune system, T cells and B cells, that if any of you paid attention to COVID over the last 18 months will appreciate these are the key cells that help fight viral infections, but also all these other white blood cells that fight bacterial infections. But these stem cells also give rise to red blood cells that carry oxygen around your body and platelets that help your blood clot and stop you bleeding. Now, I'm going to change tack here because I also look after patients who have very rare diseases, and these are called primary immunodeficiencies. These patients are born without a normally functioning immune system. And this picture here, these pictures here are of a young boy called David Vetter, who was also known as the bubble boy. He was born with a completely absent immune system. And it was a disease that at the time had only very recently been understood or diagnosed. It's a disease called SCID, or severe combined immune deficiency. His only way of surviving without dying of infection was to live in a bubble. And I find this quite heartbreaking. This young boy, as a baby, lived in a bubble, and he never had any contact except through these portals where people would put their arms, but they were separated by plastic. And he grew up living in this bubble. And when he got a bit older and was increasingly frustrated, a spacesuit was made for him so that he could venture outside, but still be in his own environment. Eventually, by the time he was about 12, his parents decided that his quality of life was appalling. And he was one of the first kids in the world to undergo stem cell transplantation for his underlying immunodeficiency. And at that time, stem cell transplantation wasn't very successful. We hadn't worked out how to do it very well. And he sadly died about a year after his transplant of complications. Patients with inherited or primary immune deficiencies are not able to fight infections. So this is a classic doctor talk. Here's some pretty disgusting, gory pictures for you. So this is a picture of a foot of one of my patients who has a non-functioning immune system, and these are his verrucas on the bottom of his foot, which are caused by human papillomavirus. So when my kids moan that they've got a single verruca on their foot and I just ignore it, this is the reason why. This is what I call a serious case of warts. On the other side, you can see a chest X-ray, and the black bit is the normal lung tissue. And this white area here is basically a hole which is full of fungal infection, and it's called an aspergilloma. Now, people with a normal immune system don't normally get fungal infections because our immune system can fight them. Other patients with abnormal or mutations in genes that help their immune cells function develop complications where their own immune system actually tries to attack their own normal tissues. This picture here is an elbow, and this is a granuloma, and this looks absolutely disgusting. But this is the result of immune cells thinking there is an infection there and being continuously activated and trying to destroy the normal tissue. But there aren't actually any bugs there at all. It's completely sterile. And this picture here is a scan of a patient who's got inflammation of the wall of their major arteries. And this is the aorta, and you can see where it's lit up. That's inflammation, and that's the patient's own immune cells attacking their blood vessel walls. And finally, patients with immune deficiencies are much more likely than normal healthy people to develop cancer. And this is because cancer cells acquire mutations that make them behave differently to normal cells, and they look different to normal cells. And remember that our immune cells are designed and trained to recognize any cell in our body that looks different or looks potentially dangerous, and they're designed to kill them. So if you don't have a normally functioning immune system, there's a good chance that cancer cells will develop more readily without your immune system noticing. Most immune deficiencies are caused by specific genetic mutations that patients inherit from their parents. And so that's an alteration in the nucleotide sequence, so the ATGC sequence that makes up the genetic code. And some of these rare diseases are caused by a single nucleotide, so a single letter change. And this can result in no protein being made, or abnormal protein being made, or a protein being made that doesn't work properly or is overactive. And the genes involved in immune deficiencies are normally switched on and expressed in immune cells that develop from bone marrow stem cells. So, when we consider the clinical use of gene therapies and stem cell therapies, I'm going to focus on inherited diseases, but also on cancer. But you should remember that not all diseases are inherited, and at the moment, not all diseases can be treated by stem cell transplantation or gene therapy. The most commonly used stem cell therapy is stem cell transplantation or bone marrow transplantation. And we use this to treat or cure various blood cancers like lymphoma or leukemia. And we also use them to treat and cure immune deficiencies where the genetic mistake occurs in a stem cell in the bone marrow that gives rise to the immune cells. So where do we get hematopoietic or the adult stem cells from? Well, we can find them in the blood of patients and collect them on a machine, a bit like a dialysis machine called an apheresis machine. We can find them actually within the bone marrow cavity itself. So this is a bone marrow harvest where a patient has a general anaesthetic and big needles are driven through the cortex of the pelvic bone, and the thick gloopy bone marrow is sucked out. And when we do a bone marrow harvest, we take about 1.5 litres of bone marrow cells from the patient's pelvis. This is remade very rapidly. The other source of hematopoietic stem cells is the umbilical cord. So you can see this rather disgusting looking placenta here, but the umbilical cord, which is this slightly dull purpley looking bit here, is enriched with hematopoietic stem cells. Bone marrow transplants or stem cell transplants, which we do a lot of in our hospital, is a very risky procedure. Patients are in hospital for up to 6 weeks, and before we give them healthy stem cells from a donor, we have to kill off their own stem cells in their bone marrow. And their own stem cells may be contaminated with leukemia cells or lymphoma cells, or their own stem cells may carry a genetic mutation in the case of immune deficiency. And we kill their stem cells off by giving the patient high doses of chemotherapy and sometimes radiation therapy. And we basically kill off all the cells in their body that are able to make blood cells and able to make immune cells. So during the period after we've infused healthy stem cells from a donor, it takes up to 2 weeks before those new stem cells find a niche in the bone marrow space and are able to produce healthy new blood cells and immune cells. And during that time, the patient has no immune cells at all and doesn't make any blood cells. So if the transplant doesn't work, if the donor's stem cells don't take, the patient will die because it's not possible to survive without healthy blood cells or healthy immune cells. So this is a risky procedure. The lowest risk of dying from a transplant is about 10%, and the highest risk is about 40%. So these are very serious undertakings. So are there any alternatives? Over the last couple of decades, we have been developing gene therapies to correct and replace missing genes. And more recently, we've been developing genetic engineering to alter and improve immune cells so that they can fight cancer cells, and more of that in a minute. So gene therapy is any treatment that introduces an extra healthy gene into a cell, and it replaces existing non-functional genes. And I say here it's like a kidney transplant, basically, because when you have a kidney transplant, the surgeons plumb in a new healthy kidney, but they leave your old one in place. So in this case, in gene therapy, we give you an additional copy of a healthy gene, but your cells will also have the mutated gene still present. Gene editing is different because it actually specifically repairs and replaces an existing abnormal gene. Gene therapy can alter what we call somatic cells. These are mature cells in the body, like blood cells, bone cells, heart cells, liver cells, or immune cells. Or they can alter germline cells, which would be sperm cells, egg cells, or the very early embryo. The only reason I mention this is because there are fundamental differences in the risk. If you do gene therapy or gene editing on a somatic or a mature cell, that change in the genetic sequence cannot be passed on to anyone other than the patient. Whereas if you do gene therapy or gene editing on a germline cell, if the patient then becomes pregnant or makes someone else pregnant, that change in the genetic sequence can be carried on into the next generation. So, final couple of examples. All gene therapy at the moment is called somatic gene therapy. So all gene therapy is on mature cells in the body. And this is an example where we can correct a patient with immune deficiencies' own stem cells by introducing a working copy of the gene into their stem cells. And we do this in the laboratory. And actually we use specially designed viruses that deliver the gene into the patient's cell. The virus has been disabled, so it can't replicate or cause an infection, but it uses its ability to enter another cell and deliver its genetic material to the cell. And the genetically modified cells, which are shown here, are then infused back into the patient. So it's an alternative way of doing a stem cell transplant, but instead of using a healthy donor's cells, we use the patient's own cells, which we've corrected in the laboratory so they express a healthy gene. Another form of gene therapy that we use frequently now in the hospital is the use of genetically engineered immune cells. We take immune cells, in this case a T cell or a T lymphocyte from a patient with cancer, and we genetically engineer that T cell so that it has we introduce a gene that encodes a protein receptor, which goes to the surface of the T cell and allows that T cell then, after it's been modified, to recognize and kill cancer cells. So it's basically like equipping the T cells with a weapon that allows them to recognize and kill cancer cells. And you can see in this picture here that these white cells are the T cells and this pink cell is a cancer cell. So after we've genetically modified the immune cells, they're able to recognize and kill cancer. So we then infuse those back into the patient. These cells are absolutely amazing. This was the first ever patient treated. This is a girl called Emily Whitehead who received these CAR T cells in April 2012 in Philadelphia in the US, and she's still alive. And she'd been given less than 2 weeks to live when she had a single infusion of these immune cells. What you can see here are some scans, and this is called a PET scan, and the bright red blobs is tumor because that's metabolically active cells. And you can see before and after a single dose of these genetically engineered immune cells or CAR T cells, the shrinking of the tumor. And in fact, this red blob here is the heart and these are the kidneys. So none of the tumor is left behind. They're fantastically effective, but they're extremely expensive. Currently about £30,000 for a dose, a single dose, but many patients only need one dose. In the last few years, there have been examples of using gene editing in patients. So you might have heard of gene editing or CRISPR-Cas9. It's an enzyme. Cas9 is an enzyme, and there's guide RNA. So it's a template of RNA, and the enzyme and the RNA come together and they can localize to a very specific site of DNA, and they allow you with absolute precision to cut out and repair a piece of DNA within an individual gene within the whole genome. And this is a picture here of the Cas enzyme, and the yellow bit here is the guide RNA, and you can see that that is binding to the unraveled strand of the double-stranded DNA, and it's telling the enzyme where to cut the DNA. The first human who received gene-edited cells was a little girl treated at Great Ormond Street Hospital in November 2015. Editing in the germline of embryos or sperm cells or egg cells is not yet being done in human beings. Some of our colleagues are developing strategies to use gene editing to understand why embryos miscarry, and they're doing these experiments in mice at the moment. So finally, when we think and look into the future, and this is going to be your future, what do you think we as individuals and society need to think about? We need to consider what are the risks and implications of somatic versus germline gene therapy and gene editing, and whether there are good or bad uses of gene therapy, and if there are, can we distinguish between them? I think most people are happy to use gene therapy and gene editing to correct disease and to make people healthy or to prevent people dying from cancer. But what about performance enhancing gene therapy? Or what about correcting traits like the colour of your eyes or the colour of your hair? And also, how will it affect our acceptance of people who are different, for example, deaf people or blind people? Will it make us less tolerant? And the gene therapy that we use for immune deficiencies are costing about £1 million per patient. And the gene therapies being used in cancer patients are about £300,000 per patient. So can we afford this, and will it only be available to the wealthy? So I'm going to finish there. and I'm going to stop sharing my screen if I can.
Thank you. That was really great. That was a really good lecture.
Thank you.
So, we now have a few questions to ask you from our attendees, if that's okay.
No problem.
So you mentioned the COVID-19 vaccine and just vaccines in general, and someone wanted to know, how does the vaccine work? If you could just give a quick sum up.
Oh yeah, okay. So one of the other things I do is I'm the Director of Infection and Immunity at UCL. So I've had quite a busy year with the whole COVID thing going on. So vaccines work by basically duping your immune system into thinking that you've been infected by the virus. Okay, so what they do is they either inject into your body little fragments of virus protein, but not the virus itself. So the virus can't divide, it can't cause an infection, but they give little fragments of the protein, and your immune cells see these bits of protein and get activated. And your immune cells, when they're activated, divide, and you suddenly, instead of having 1 or 2 immune cells which are like soldiers, you suddenly get a massive battalion, and they're all able to recognize that virus. And there are different types of vaccines. Some of them, like the one I talked about, the Pfizer vaccine, is an RNA vaccine. Others of them actually are protein-based vaccines, and they provide peptides or proteins but they're there to just dupe your immune system into thinking that you've actually had the infection, and your immune system tries to fight it. And the good thing about your immune system is that these cells then survive sometimes forever. So you will have all had things like chicken pox as a kid, most of you, and you will still have immune cells in your circulation that can recognize and kill chickenpox virus if you were ever to encounter it again.
Yeah. Also regarding vaccines, what would you say the potential problems of using the Pfizer RNA vaccine are compared to a normal inactive pathogen vaccine?
Oh, I don't really think there are any problems actually, because all the cells in your body are designed to make protein and use ribosomes to make protein. So I don't think there are any potential problems with the RNA vaccines. One thing I would say, however, is that the RNA is packaged in little lipid nanoparticles, and there are exceptional cases where people can have reactions to that, but it's been very low.
So away from vaccines and stuff, what can stem cells not turn into? So you mentioned all the things that they can potentially divide into, but what can they not?
Oh, I don't know the answer to that. I mean, I think if you're an embryo, if you're an embryonic stem cell, you can literally turn into anything. If you're a hematopoietic stem cell, you can't turn into a liver cell or a heart cell, but you can turn into a T lymphocyte or a B lymphocyte or a dendritic cell or a neutrophil or a red blood cell or a platelet. So yeah, adult stem cells are more restricted in what they can become.
So it depends on the type of stem cell it is.
And the iPSCs, the induced pluripotent stem cells in the lab, which are being used in research, depending on which cocktail of growth factors you grow them in, they can become all sorts of different things.
Yeah. Also, okay, so in order to aid in inhibiting the production of viral proteins, do you think it is possible to use a selective chemical tag, such as altering the genome of the virus to code for solely stop codons? Because they feel that this would provide a general solution that is non-specific to any singular viral genome, and then perhaps protecting against viral mutations. So, and then if you do think that's possible, what disadvantages or practical issues would this have? It's quite an interesting question.
Blimey, that's a difficult question. Can you say the first bit of it again?
Yeah. So do you think that it's possible to use selective chemical tags such as altering the genome of the virus to code for solely stop codons?
Oh, so I guess in theory you can do all sorts of things like that, but if you were that would be useful if you were going to use the virus as a vector to deliver genes. But if you wanted to reduce the pathogenicity of the virus, you would have to get all the virus that's out there in the community and isolate it from patients and alter it. So I'm not sure how you could actually make that happen.
Yeah. Maybe one day one of these people who's attending.
Exactly. I may be missing the point. If I am, anyone can email me, that's fine, and I'll answer.
Yeah. So do you think there's any way of enhancing the immune system of a person who has a weakened immune system, or it just doesn't have an immune system?
Yeah, so the way we do that is through gene therapy. So if patients are born without a specific gene that helps their immune cells to either develop in the first place or to function correctly, we can replace that gene using stem cell gene therapy. And we're doing that at the moment for patients with 5 or 6 different types of inherited immune deficiency. And the other way of doing it is the more, I guess, brutal way, which is to do a stem cell transplant, and rather than just correcting the genetic mistake, actually just replacing all of the stem cells with healthy ones from another person. So you can do that, yes.
What are the limitations of stem cell therapy, and what would you say went wrong with the case that you mentioned, the David Vetter?
Yeah. Okay, so when that was done, the way back in the early 1970s, the way stem cell transplants were performed, doctors then used very high doses of chemotherapy and radiotherapy, which actually were quite toxic. So they not only killed the cells in the bone marrow, but they were damaging to the heart and the lungs and the kidneys. So patients had side effects like that, and sometimes the chemotherapy was so toxic patients would die from that. The other things that happened was that we weren't very good then at matching a donor and a recipient very well, because we didn't really understand all the things we know now about how immune cells recognize cells from another person. So our ability to work out whether someone's a really good match or not for a transplant is massively better now than it was then.
Yeah. Why do you think it's difficult to instruct stem cells to differentiate into specific specialized cells?
Because they have to have a very they have to grow in a very precise cocktail of growth factors, which basically are hormones or enzymes or ligands, which trigger particular pathways within the cell, which in turn switch on particular genes in the cell, which tells the cell to become a liver cell or tells the cell to become a skin cell. So for example, a keratinocyte makes keratin. It's got a tough outside. It can't contract, it can't move, whereas immune cells have a skeleton that allows them to sort of travel around the body. So all different cells have different proteins, and that's all controlled by which genes are switched on and off at a given moment. And those are controlled by things called promoters, that are sequences that switch on and off proteins, and some of them switch off at different bits of the cell cycle or on at different bits of the cell cycle, and some of them switch on and off depending how mature the cell has become. And all of these things can be influenced by the surroundings of the cell, so the growth factors it's in and what other cells it's sitting next to. It's pretty complicated. It's what is so amazing I think, is that in our bodies, our liver basically contains liver cells, and our heart contains heart cells, and our skin contains skin cells, and they don't get mucked up. You know, they don't go to the wrong place all the time, or our skin doesn't start suddenly becoming bone or something. I mean, it's,
I don't often think about that, but it's really amazing.
it is amazing. And when you think how many cells turn over every day,
Yeah.
you know, they don't make many mistakes, actually. They're really pretty good at copying the genetic sequence. I think it's amazing. We get less good at it as we get older.
Yeah. Do you think stem cells can stop autoimmune diseases by potentially replacing the immune cell that attack the normal cells?
That's a brilliant question. So basically, we use, for some types of autoimmune disease, we use stem cell transplants, but we use the patient's own stem cells, and it kind of resets their immune system a bit. And it's not fully understood how that works, but definitely for some very severe autoimmune diseases where immune suppressing medicines don't work, we can use stem cell transplants for that.
Yeah. So do you think there's a difference between stem cells derived from humans, plants, and animals if it was used for stem cell therapy?
Do I think, say that again?
So would there be a difference between stem cells derived from humans, plants, and animals in terms of using it for stem cell therapy?
Oh, yeah, good question. I don't know. I haven't done any stem cell work in plants, but in yes, I mean, mammals would be the same.
Yeah. So you know how you mentioned stem cells and how when we age it slows down in terms of our cells dividing. So do you think stem cells and gene therapy play a role in slowing down the age cycle? Can they play a role in slowing down the age cycle now in the near future?
Oh, that's a good question. I don't know. So it's slightly out of my comfort zone answering a question like that. But I would say, I don't know whether you learn, for those of you who are doing A levels, I don't know if you learn about telomeres, and these are sort of caps on the ends of the genetic material that basically, as the cell keeps dividing, they get shorter and shorter and shorter. And they basically give an idea of how old the cell is and how long it can keep dividing and renewing itself. And as we know, as we get older, that we're less good at repopulating cells in our body and our tissues. Whether there will be ways where we can rejuvenate our stem cells by using the sort of things that are being done in research labs at the moment to generate new stem cells, I guess it's possible. I mean, you know, I don't know whether people really do want to live forever. I don't know whether I really do. I'm a bit, you know, I'm only 53, but I'm quite tired. I don't think I want to still be here in 100 years' time. Yeah. But yeah, I don't know.
Do you think there's any way of taking adult stem cells which can be a painless procedure?
Oh yeah. So one of the things I showed you was the stem cells in a bag above a machine, which I called was an apheresis machine. You take that from the blood. So you basically, you give a patient or a donor 5 days of injections, just a tiny injection under the skin, a bit like an insulin injection or a blood thinning injection, and it's a growth factor called GCSF, and basically it makes the stem cells that are usually stuck in the little niches inside the bone marrow detach, and so that they can they sort of can free up and they circulate through the bloodstream, and you can collect the stem cells out of a vein. So you have a little cannula in one arm and you have a cannula in the other arm, and it goes through a machine and it goes past through a centrifuge, and a laser is shone through it, and the cells are separated by their density, their size, and their ability to scatter light. And you can separate all the different cells of the blood and collect, I don't know how the machine works, to be honest, but you can collect the stem cells in a bag, and all the other cells go back in the tubing back into the patient in the other arm. And you basically, you have to have a needle in each arm, but they're small, and you have 4 injections, and you have to sit on a bed watching the telly for about 4 hours, and you can collect enough stem cells to do a whole stem cell transplant. Wow. So if you are interested in being a stem cell donor for the Anthony Nolan, that all you need to do is to get on the register, is give a swab from inside your mouth, and if you were ever called up to save someone's life, that would be all it would take. Half a day. Cool. It's very, very cool. And I would say, for anyone out there who is not Caucasian, if you're interested and you can get yourself on a register, because it's really hard for my non-Caucasian patients to find appropriately HLA matched donors for stem cell transplants. So do it if you're young and healthy and keen. I think you actually have to be 16, so I don't know how old some of you are. But go to the Anthony Nolan website if you're interested.
Have you ever seen uncontrolled mitosis of stem cells leading to cancer? And if so, how and why does this happen?
Oh man. So I guess if you mean uncontrolled proliferation of cells and division passing through the cell cycle, yes, because that is actually one of the hallmarks of cancer. So there are various different things that we use to describe what makes a cell a cancer cell compared to what makes a cell a normal cell. And one of the hallmarks of cancer is uncontrolled cell division. And that usually occurs as a result of an acquired genetic mutation in that actual cell, which means it's no longer sensitive to all the control signals or the normal regulation. And one of the reasons cancer is more common as we get older is because we acquire genetic mutations in some of our cells as we get older. These aren't things we inherit from our parents. These are little mistakes that are made every time the cell divides. When you think again about the mind-blowing how many cells have to exactly replicate every day, and they have to get their whole sequence correct. Well, in reality, they don't have to get it completely correct, because there are some shape misspellings that don't alter a protein or don't have a bad deleterious effect on the cell. But if there are mistakes that damage the cell or damage the cell's ability to function properly, then that cell, if it divides very quickly over the course of time, that will be the cell that you get more and more of it, because it's dividing quicker than the normal ones.
So I think we have time for just one more question.
Okay.
Are there any racial biases in people chosen to receive gene therapy?
No, that is a really good question. There aren't, because actually the only gene therapies available, so gene therapies for immune deficiencies or inherited diseases, are given to patients who have that genetic mutation. And in fact, because some genetic disorders, which are what we call autosomal recessive, that occur when you inherit a damaged copy of the gene from one parent and from the other parent, they are more common in consanguineous marriages. So if you have parents who are cousins, so there are some communities where that's more common, some parts of the world where that's more common. So there's an increased rate of autosomal recessive disease in some populations. So in a way, those populations often are more likely to be treated. For patients who receive the gene engineered immune cells for cancer, I would say in Europe there's no racial disparity at all about people who have access to those therapies. I wouldn't like to comment on the politics of the US health system, and it depends entirely, apart from the fact that I think it's appalling, but it depends entirely on whether you have insurance, because most of these treatments are paid for by insurance. So I think for the rare diseases, most access is through clinical trials, and if doctors have eligible patients and they can travel to a country where there's a trial open, that happens.
Yeah. So I think that's all we have. We have 5 minutes, but I think that's all we have time left for today. So thank you, Emma. That was really great. It was really insightful, and it was such an important topic to discuss and learn about. It has so many potentials and so many breakthroughs that you could have with therapeutic options. It was really interesting. Thank you also everyone who attended and for all your great questions. Really, really good questions. And then if anyone has any burning questions that have not been answered, or you would really like an answer to them, you can email the Faculty of Medical Sciences at fms.education@ucl.ac.uk, and hopefully they will have an answer for you. So really, that's it for us for this month. So thank you again, Emma, and thank you all who joined us today and attending, and we hope to see you again in the next medical science lecture.
Great.
Bye, everyone.
So everyone, and good luck if you've got exams.
Yeah, good luck.
Or assessments, or whatever they're called these days, but don't forget to still work. Okay,
Thank
bye.
you.